Welcome to our explainer. Today, we're diving into a hidden force that drives the progression of Duchenne muscular dystrophy. Think of it as a constant, smoldering fire inside the muscles, and we're going to look at one potential way to fight it. Okay, so let's get right into it. We all know Duchenne is a genetic condition, but that's not the whole story. There's this other process going on, a kind of cellular stress that's constantly fanning the flames of muscle damage. So how are scientists trying to put that fire out? You know, in a healthy body, inflammation is actually our friend. It's a short, sharp response to an injury, it helps us heal, and then it goes away. But in Duchenne's muscular dystrophy, well, that's a completely different ballgame. What happens is the inflammation switch gets flipped on, and it never flips off. It becomes chronic. The body is in this constant state of high alert. And over time, that response stops being helpful and actually starts causing more damage, leading to scarring in the muscle. So the big question is, what's lighting the match in the first place? To find the source of this fire, we have to get really small. I mean, we need to zoom way, way in, down to the microscopic power plants that are inside every single one of our muscle cells. And here's where it all starts, the mitochondria. You know, these are the engines of our cells. And what's so wild is that research shows these engines start malfunctioning really early on in Duchenne, even before the major muscle breakdown really kicks into high gear. This image here really maps out the chain reaction perfectly. You see those damaged mitochondria at the bottom? They start sputtering and releasing these harmful molecules. And if you follow the arrows, you can see those molecules directly flick on a major inflammation switch in the cell, basically shouting emergency and calling in the full immune response. So what are these harmful molecules? They're called reactive oxygen species, or ROS for short. The best way to think about them is like dangerous sparks flying off a faulty engine. They're super unstable and they just bounce around the cell, damaging everything they touch, DNA, proteins, you name it. And they are the things that directly trigger those inflammatory alarm bells. And this is the absolutely crucial part. It creates a devastating, vicious cycle. The damaged mitochondria make ROS, which causes inflammation. That inflammation then circles back and causes more damage to the mitochondria, which then, you guessed it, pump out even more ROS. To stop the damage, you have to break the cycle. You have to find a way to put out those sparks. So that brings us to a potential solution. If the problem is these dangerous cellular sparks, what you really need is a fire extinguisher, right? Well, in the world of our cells, that's exactly the job of antioxidants. The concept here is actually pretty simple and elegant. You've got the problem on one side, all these unchecked ROS molecules causing chaos. And then you've got the solution, antioxidants. Think of them as cellular peacekeepers. Their entire mission is to hunt down and neutralize ROS before they can do any harm. So by swooping in and scavenging all these sparks, antioxidants could, in theory, break that vicious cycle we were just talking about. Control the ROS, and you can dial down the inflammation, which in turn helps protect the mitochondria from getting more damaged. It sounds great on paper, but does it actually pan out in the real world? Let's take a look at a case study. Okay, we're going to zoom in on one specific synthetic antioxidant. It's called Idebenone, and it's been studied in Duchenne patients specifically to test this exact theory. So what exactly is this stuff?
Well, Idebenone is a man-made antioxidant. It's basically a souped-up version of coenzyme Q10, which you might have heard of. It was engineered to do two key things. First, to be a really potent scavenger of those ROS sparks. And second, to directly support the mitochondria and help them produce energy more efficiently.
This quote here is from a really important long-term study called SYRS. What researchers did was track Duchenne patients who were taking Idebenone for up to 6 years. This gave them a super rare and valuable look at the long-term effects of using this antioxidant strategy. And the study focused on something absolutely critical in Duchenne, breathing. As the respiratory muscles get weaker, breathing becomes one of the biggest challenges. So the million-dollar question for the researchers was simple. Could an antioxidant like Idebenone actually slow down this decline? And the headline result was 50%. That is a huge number. The study found that taking Idebenone long-term cut the annual rate of decline in breathing function by half. Basically, patients' breathing capacity was declining 50% slower while they were on the drug compared to the period before they started. And the good news didn't stop there.
This wasn't some one-off effect. It was consistent year after year. And maybe most importantly, this translated into real-world benefits. Patients had fewer serious lung infections and fewer hospital visits because of breathing problems.
Okay, so those results are, wow, they're incredibly encouraging. They show that going after these secondary effects of the disease, this hidden fire of inflammation, can make a real meaningful difference. But, with any good science, we have to pull the camera back and look at the whole picture.
So first, the big takeaway. The strategy itself, using powerful antioxidants to fight the downstream chaos caused by the lack of dystrophin, it seems to be a really valid and promising approach. The CIRRUS study is strong evidence that this can protect vital functions like breathing.
But, and there's usually a but in science, right? It's also important to be honest about where the research stands as a whole. When a group of scientists did a huge review looking at multiple studies on antioxidants for Duchenne altogether, their conclusion was that the overall certainty of the evidence was still considered low.
Now, that does not mean the CIRRUS study is wrong. Not at all. It just means the scientific community is, by its nature, very cautious. What they're saying is, hey, this is a really, really powerful signal, but we need more data to be absolutely sure. Promising results from one study, even a great one like CIRRUS, need to be backed up by more, larger trials to build a rock-solid case. And this really leads us to a fascinating final thought about the future of treating complex diseases like this one. Maybe the ultimate answer isn't a single silver bullet. Could it be a cocktail of therapies, a combination where one treatment goes after the root genetic problem while another works right alongside it to put out that secondary inflammatory fire? That might just be the path forward.